Life Sciences Week 4 Video Lecture 7, Organic Compounds, Proteins. Hello class, we are taking a look at proteins here. So we just finished looking at carbohydrates. Let's look at our objectives. 2 nice, clean, simple objectives. That's nice. So they are describe the structure of proteins and list and give examples of proteins in the body. I think we can do that. All right, so this is not a protein. This is Linus Pauling. Linus Pauling, what does he have to do with proteins? So Linus Pauling was an American, let me think, what is his job title? A theoretical physical chemist is what he was. And so he was doing his work in the early 1900s through, I think he died like in the 80s, in the 1980s, or maybe even later. But he did a lot of stuff like in the 1930s, 20s maybe even, in 30s and 40s and 50s and 60s, and had a long career, not just in chemistry but also in activism. He won 2 Nobel Prize, one for peace in his resistance to creating nuclear warfare, and the other in chemistry. So in chemistry, it may have been chemistry, but it had to do with his work in chemistry. I don't know exactly what it was. Anyway, but he's worth looking up. Quite a list of accomplishments. But what does Linus Pauling have to do with proteins? Linus Pauling is a chemist who did a lot of work with coming to understand, before we could really see them at all, coming to understand the structure of complex organic compounds. So his work contributed to what Watson and Crick would eventually discover as the structure of DNA. And really a lot of what he did was he figured out the structure of large complex proteins, or I should just say complex proteins. And so the work that he did, to do that, he used X-rays. He used a technique, I think, that was developed in Germany, although I could be wrong about that. But he at least learned about the technique while he was there. And it involves using X-rays, and that these microscopic molecules have specific X-ray signatures, and we can study the signatures, and that can help us to sort of figure out the physical structure of these large complex organic compounds. So so one of the things I think is cool about that is that builds on the work of the Curies. So remember, in chemistry, Marie Curie and her husband, oh my gosh, what's his name? Louis, maybe? Oh gosh, it escapes me, sorry. But anyway, the Curies, they essentially developed X-rays, and so it was, kind of building on their work, that Linus Pauling and others who did a lot of work around discovering the structure of complex organic compounds. Is where their work came from. So later in Linus Pauling's life, maybe you recognize his name as somebody who strongly promoted megadoses of vitamin C, and he and his colleagues did a lot of work on that, on the structure of vitamin C and the uses of it in the body, and potential protective functions that it might have against cancer. So anyway, that's Linus Pauling and his contributions to the understanding of the structure of complex organic compounds, such as proteins. And so that takes us into looking at proteins today. So the protein we're looking at here is hemoglobin. So although hemoglobin isn't an enormous protein, it is a complex protein. And so we see many layers of structure within a hemoglobin protein. So what we're looking at here on the left is a red blood cell. Inside a red blood cell, so red blood cells are cells that are specialized for carrying hemoglobin molecules. And hemoglobin is a protein, and its function is to, well, you can see in it has these 4 iron molecules embedded within it, and those iron molecules create the receptor site where hemoglobin binds oxygen. So hemoglobin is how red blood cells carry oxygen. We have something like 280 million hemoglobin molecules per red blood cell, and each hemoglobin molecule carry 4 oxygen molecules. So that gives us a nice oxygen carrying capacity in our blood, as long as we're making hemoglobin correctly and red blood cells correctly and all of that. So anyway, it is a fantastic protein for us to look at to get a sense of how complex proteins are. So we know that proteins, all proteins, are made out of amino acids, and the amino acids are bonded to each other through dehydration synthesis reactions. So those are the reactions we looked at when we were looking at carbohydrates, how we can have 2 neighboring molecules and we can pull a water out of them collectively, in a sense. So take a hydrogen from one and an oxygen and a hydrogen from the other and create a water molecule, and in that process bond those 2 neighboring larger molecules to each other. That's a dehydration synthesis reaction. So that's how amino acids attach to each other to make a protein. Let's take a look at the structure of an amino acid. So we have 20 different amino acids in our body. 8 of them are considered essential amino acids. So what that means is that those are amino acids that we cannot make in our body, and the other 11 are considered non-essential amino acids. The non-essential ones, we still absolutely need them to be able to make all of our proteins, but they're considered non-essential because we do have the capacity to make them from scratch in our cells. So that's the difference between essential and non-essential amino acids. But any amino acid has this basic structure that we're looking at here. And we saw this structure when we were looking at amino acids as buffers. So we saw that on the left, so the way this is pictured, on the left side of this amino acid, we see the amino group. So that's the nitrogen with the 2 hydrogens. On the right side of this amino acid, we see the carboxyl group. So that's the carbon that's more to the right, and the 2 oxygens that are attached to it, and then the hydrogen that's attached to one of those oxygens. And then we have that central carbon, and that central carbon has 2 things attached to it. One is a hydrogen, and you can see the way that this model is built, or the way that this molecule is being depicted here. That hydrogen that's attached to the central carbon is supposed to look like it's like going away from us. That's giving us sort of a 3-dimensional kind of feel to this molecule. So that hydrogen is off behind, and then what's coming out toward us in the front is it just is represented with an R, and that's the side group. So remember that with amino acids, that side group is different for every single different amino acid, and that's what gives each amino acid its slightly different qualities and different behaviors. But every amino acid has the amino group and the carboxyl group around that central carbon, and a hydrogen on that central carbon as well. That structure is the same for every amino acid. Okay, so how do amino acids attach to each other? First of all, let's look at all these amino acids. So this is a chart of all 20 amino acids, and the way that they are shown on here, if we zoom in on one, we can see that, so if we look at the one on the top left, we see an OH and a line, and then a double line and an O. So that's the carboxyl group of this particular amino acid, and then we see a line and then it turns to the right, and then we see NH2, so that's the amino group. So these amino acids, as they're shown on this chart, are oriented differently than the one we were looking at on the previous slide. So it's like we've taken the one from the previous slide and rotated it counterclockwise, like 90 degrees-ish or so. So then what that means is the part that is coming down is the side group. And so we can see that each of these amino acids has a different side group. So we can see nitrogens and hydrogens in some of these side groups. And remember, what we're looking at here are representations of organic compounds. So remember that organic compounds have carbon as a base kind of building block atom. And so with all of these, when we see these lines, either hexagons or pentagons or zigzag lines, at every corner, if it doesn't have some other letter like an N or an O, at every corner or every end of every line is, it's implied that there's a carbon there. So even though we don't see any letter C's here, there's carbon all over the place here because they're at every single corner. They're the basic building block. Because remember that carbon atoms can form 4 covalent bonds, and so it makes it a very sturdy molecule to build sturdy atom to build molecules off of. So here's some different amino acids. These, the two that we're looking at here on the right, there's a negative charge to their side group. The one on the left here, there's a positive charge to the side group. So you can start to see that some of these amino acids are going to behave a little differently in given situations if some of the side groups have different charges to them, if some of the side groups are larger or smaller, some have nitrogen, some are just carbon, and some have sulfur in them. So there are variations. Let's look at the bottom left here. Alanine is our smallest and simplest amino acid. So off of that central carbon, all we have is another carbon. It's that's as small and simple as it gets as far as side groups. So anyway, so this gives you a little view of the difference of between one amino acid and another, and the similarity between one amino acid and another. So let's take a look at how amino acids bond to each other. So up on top of this chart, we see 2 neighboring amino acids. So on both of them, we can see that central carbon in the middle, and actually this is not showing side groups. It has hydrogens instead of a side group, but that's okay. That's not the important part here. The important part is, let's orient first to the amino acids that we're looking at. So the 2 amino acids on top, what we see, the one on the left, they're essentially identical to each other side by side here. So the one on the left, we see the central carbon, we see the amino group on the left, we see the carboxyl group on the right. And then exact same thing for the top right amino acid. We see the central carbon, we see the amino group on the left and the carboxyl group on the right. So if you see the red OH on the left-hand amino acid and the red H on the right-hand amino acid, those 2 hydrogens and that oxygen are going to come together to form a water molecule. So that's the water molecule that will form in this dehydration synthesis reaction that occurs. So we're going to take that water molecule out, and then what happens is the carbon of the carboxyl group on the left bonds with the nitrogen of the amino group on the right. So now we have a covalent bond between that carbon and that nitrogen. That particular bond between 2 amino acids we call a peptide bond, and it is peptide bonds that form proteins, because at the most basic aspect of the structure of a protein is a string of amino acids. And so those amino acids are strung together through these peptide bonds, which form through dehydration synthesis reactions that then bonds together the carbon of the carboxyl group and the nitrogen of the amino group. Okay, so that's what a peptide bond looks like, and where and how that forms. So amino acids, let's see, do we have a list of them here? No, we just have their structure. Okay, so what we're looking at here is the sort of layers of structure that we have that make up an amino acid. So the structure that I was talking about, the string of amino acids, that's called the primary structure, and we can see that on top here. So the primary structure of an amino acid is the sequence of a protein, is the sequence of amino acids. So which amino acid gets bonded to what's the first amino acid, what's the second amino acid, what's the third amino acid, on down through the whole growing peptide chain here. So that's the primary structure, the sequence in which the amino acids occur. So very small proteins might just have primary structure, like if they're 5 or 6 amino acids long or something like that. Generally proteins are bigger than that, however, and so when they get bigger than that, they start to form secondary structure. So here on the what's currently on the bottom left of the screen, we see secondary structure. So as the chain of amino acids grows, and so let me say a little aside here that amino acids are formed inside our cells by organelles called ribosomes. Ribosomes are kind of they're a very basic organelle that we find in plant cells, in animal cells, in bacteria cells. They're one of the most primitive organelles. In cells that, like bacteria, that that don't even have a nucleus. Because they're very, primitive isn't the best word, but they're a much simpler system than any one of our cells. They still have ribosomes. So which means they have the ability to make proteins. So anyway, that's where this is happening. When we're building proteins, we're doing it inside any one of our cells in our whole entire body. We should be able to make proteins, and it's the ribosomes that are doing it. So anyway, the ribosomes are assembling this sequence of amino acids. Once that sequence of amino acids starts to gain a certain amount of length, it will start to twist, and so there are a couple, and basically based on which amino acids are in sequence with each other, we're going to get either an alpha helix or a beta sheet. So those are just two different shapes that amino acids can take, kind of based on how the amino acids in sequence are attracting each other or repelling each other, so how they're interacting with each other based on what those side groups are. So we'll start to get this secondary structure. So we'll get these twists, and this is one of the things that Linus Pauling discovered is like how many amino acids are in a sequence for each turn of an alpha helix, for instance, like that kind of thing. Okay, so let's keep looking at the structure. So we have our primary structure of our sequence of amino acids. We have our secondary structure, which are the twists and the kind of bends and stuff that begin to form in this growing chain of amino acids. So it's kind of like in this top picture, we're looking really closely at it so we can see the individual amino acids, but then we pull out a little bit and look at it from a little farther away, and we can see the coils and the twists and stuff. So, but there's more. So the next thing we have is called the tertiary structure. So the tertiary structure is where the different coils, so the alpha helixes and the beta sheets, those different things begin to interact with each other. So there are folds and twists and turns where perhaps part of the alpha helix will be attracted to part of the beta sheet, and that will hold them in relationship to each other. So maybe they're side by side, or they're at perpendicular angles to each other, or whatever. But we form a very distinct three-dimensional shape, again, based on how the side groups of the amino acids are interacting with each other. Are they repelling each other? Are they attracting each other? Are they creating a straight line? Are they creating a curved line? Things like that. So that gives us our tertiary structure. And then, so some amino acids, all they have is their secondary structure. So if they're kind of smaller, they'll just have primary and secondary structure. Moderate to large proteins are going to have tertiary structure. And the most complex of proteins have what's called quaternary structure. And so that structure is where we take like 2 or 3 or 4 different completely formed proteins with tertiary structure and put them together. So what we're looking at here, again, we're back to this hemoglobin molecule. In each of these, so we see a red portion, a green portion, a blue portion, and an orange portion. Each of those portions is a completely formed protein, and now they're all attached to each other. That's our quaternary structure. That's the most complex level of structure. Now we'll see in some of our examples that we can get more complex than that if we need to sort of, build a big significant rope in our body out of protein or something like that. But this gives us an idea of the basic structure of proteins. So let's see, what else do we need to say about proteins? Okay, let's talk about breaking proteins down. So we've talked about building proteins. And let me mention one more thing about building them. So the secondary and tertiary structure are held in place by hydrogen bonds. So remember that hydrogen bonds, I was referring to that without calling them that, but I was saying that certain parts are attracted to each other, other parts are repelled from each other. And so the parts that are attracted to each other form hydrogen bonds. So we know that hydrogen bonds are formed between slightly positive and slightly negative ends of neighboring molecules. And so that's what we have here that holds the secondary and tertiary structures in place. So those hydrogen bonds just form naturally as a part of building a protein. So what happens if we want to take apart a protein, which we need to do anytime we're eating a protein? So let's look. I had mentioned building proteins inside our cells. Well, let's look at it from the other direction. Let's say we've eaten protein and now we want to take it apart. So if we want to take apart a protein, we need to disassemble it all the way down to the individual amino acids. But how do we get there? The first step is we break the hydrogen bonds. And where and how does that happen? We break hydrogen bonds in proteins by exposing them to a strong acid. And so in the case of our bodies, that's hydrochloric acid, and the very low pH of our stomach. So that low pH takes apart those hydrogen bonds, and so all of the secondary and tertiary structure goes away, and then we have just these long strands of amino acids that then our protein digesting enzymes can start to snip apart into smaller chains of amino acids. So those smaller chains of amino acids are called peptides. And the protein digesting enzymes that we have in the stomach and also produced by the pancreas that work in the small intestine, as a group they're referred to as proteases because they break down proteins. But what specifically would they be doing? Well, if we formed a peptide bond through a dehydration synthesis reaction, then we would break a protein through hydrolysis, or break a peptide bond through hydrolysis. So we insert a water molecule in there. The hydrogen goes to one side, the oxygen and other hydrogen go to the other side, and that separates two neighboring amino acids, so that breaks the peptide bond. So we're going to break them the exact opposite way as we formed them, and that's what the digestive system does very well. And so once we get them down to individual amino acids, then we can absorb them into the bloodstream. We can then deliver them out to cells in our body, and then the cells can take them up and use them. And guess what our cells are going to do with amino acids? They're going to reassemble them into our very own proteins. So so we may be eating proteins that are exactly like proteins that we're then going to turn around and use the amino acids to build, but we have to do it that way. We have to break them all down into individual amino acids, absorb them as individual amino acids, and then rebuild them in our cells into the proteins that we need. All right, so what else about proteins? Let's take a look at the uses of proteins in the body. So what we're looking at here is a cell membrane, and we haven't looked specifically or closely at cell membranes yet, but so if you see the sort of pinkish line that's made of kind of spheres, and there's sort of two rows of these pinkish spheres, those are phospholipids that make up the majority of a cell membrane. So we're looking at this in a line, but imagine that line continues in both directions, and toward us and away from us, so that it forms a sphere. It's like, say we took a basketball and we cut it and we're just looking at the thickness of the outside of the basketball. That's the equivalent of what we're looking at here. We're looking at the thickness of the cell membrane of a cell. But this would be, the cell membrane would continue all the way around and form like a spherical shape or whatever, some kind of shape, depending on what cell it is. So embedded within that cell membrane, we see a few proteins here. And we, just looking at these, we can kind of recognize some of these shapes. We see some alpha helixes and beta sheets in the straight lines and the twisty curves of some of these proteins. So these are smaller proteins that are embedded in cell membranes. And the small proteins of cell membranes might serve a number of functions. They may work as enzymes. They may be acting as receptors for something that the cell needs to be able to receive. They may be acting as channels for things that need to pass from the extracellular fluid to the intracellular fluid. So, there are a number of functions that proteins in the cell membrane might be carrying out. Those are just a few of them, and these are a few good examples of some smaller proteins doing that. Other smaller proteins in our body might be functioning as enzymes, but not like embedded in a cell membrane like this. Other small proteins could be acting, or smallish, moderate-sized proteins could be acting as hormones. So in the endocrine system, we have two types of hormones. We have water-soluble hormones and fat-soluble hormones. So our water-soluble hormones are made out of proteins. Okay, let's look at some other examples of proteins. So there we had been looking at proteins embedded in cell membranes, and so those are going to be smaller proteins. Also inside cells, we have small to moderate sized proteins that create structure inside cells. So that's on your notes, it's listed in the moderate sized proteins, some of these like cellular structures, such as structures that form the shapes of cell membranes, or just support the inside of cell membranes, or form specific shapes of cell membranes, like the flagella of a sperm cell, or the axon of a nerve cell, or something like that, or dendrites of a nerve cell. So those are all cell membranes that hold specific shapes, and it's proteins inside that cell, but these are going to be more like moderate-sized proteins inside that cell that mold and shape the cell membrane from the inside to hold and help to hold it in shape. Other moderate-sized proteins, are what we're looking at here on the slide. Now, these are actually proteins that are outside of cells because they're in a connective tissue, and that's the nature of a connective tissue, is that it's made up of cells and matrix, and part of the matrix is fibers. And so these protein fibers they're actually a protein called fibrin, and this is a protein fiber that forms when we need to form a blood clot. And that's what we're looking at here, is part of a blood clot. So that's why we see red blood cells kind of tangled up in these fibers. But so these are, you might call these large proteins. I guess we could call these moderate to large proteins. But you can see that they would be small enough to fit inside the cell, but in this case they're actually outside the cell, and they are creating the structure outside cells, and in this case they're creating the structure of a blood clot. So those are some examples of some moderate-sized proteins. Also, we could look at the proteins inside muscle cells that move and change shape in order to create a muscle contraction. That's another great example of moderate, and actually some of those would be definitely large proteins as well. Some other large proteins, here's a great example of a rather enormous protein. So what we're looking at here is a tendon. And it's not so much that any given protein within this is enormous, it's that it creates this huge protein complex. So if we look on the left, we see collagen. So collagen is the protein. Collagen is actually the most abundant protein fiber that we have in the body. It's a fiber that shows up outside of cells, and we find it in connective tissue. And it's very strong and does a whole lot of the soft tissue structure in our body. So we see a collagen fiber there, and then we have a bundle of collagen fibrils that create a collagen fiber. So a single collagen fibril will pair up, or more than pair, but get together with a number of them and form a collagen fiber, and then a number of collagen fibers come together to form a bundle, and a number of bundles come together to form a fascicle, and a number of fascicles come together to form so we have a secondary fiber bundle, which is called a fascicle, a tertiary fiber bundle, so we're just having like more and more of these together. So imagine looking at a rope, right? If we took a rope and unraveled it, say a rope is made up of 2 or 3 strands twisted or braided together, and if we took apart one of those strands, that is also made up of strands that are braided together. And if we took apart one of those strings, or smaller strands, we'd find more strings twisted together to form that. And that's essentially what we're looking at. If we get down to the tiniest threads, that's the protein, that's the individual proteins, but we can put them together to make these really enormous proteins that create all the soft tissue structure of our body. So So hemoglobin, actually I put in the notes as one of our large proteins. We might even call hemoglobin a moderate sized protein if we have 280 million of them inside every red blood cell. It's not so much that it's large, it's more that it's complex. But anyway, our tendons, our ligaments, our fascia, and all of that kind of stuff are, proteins upon proteins upon proteins to create these larger protein structures. And then the larger proteins inside muscles that are doing the muscle contraction. Some of the proteins inside muscles are smaller and some are definitely larger and like more complex. So there is a wide range of functions that proteins carry out in the body. In general, proteins are doing a structural kind of function, but that's not exclusively what they do. So structural that's kind of a nice thing to think about with proteins, that proteins create structure. But like if we think of some of our smaller proteins, we have, we think of antibodies and enzymes and hormones, and that certainly is not necessarily a structural function. It's more of a like an enzymatic function or something like that. But we can also use proteins for energy. So if we needed to, like if carbohydrates were scarce and fats were scarce, we certainly do all the time take amino acids, and so we'd have to break down our proteins, which we're constantly doing, of course, in our body because we're always repairing and replacing tissue. But we could break down proteins, break our own proteins down to their individual amino acids, and then some of our amino acids, depending on their structure, can be turned into glucose that we can then use to make ATP. And some amino acids don't need to be converted to glucose first, actually, and we can just put them right into the Krebs cycle and electron transport chain and get some energy out of those, the bonds of those amino acids. So we can use proteins, amino acids, I should say, as energy as well. So there's lots of uses of proteins in the body, but structural is kind of our primary use. So we talked about the structure of proteins. So we talked about how proteins have a primary structure of a string of amino acids in a specific sequence, and those amino acids are bonded to each other through peptide bonds. We talked about the secondary structure that forms once we get a certain length of that primary structure happening, so like alpha helix and beta sheets, and then hydrogen bonds holding those shapes in place, and then tertiary structure also held in place with hydrogen bonds, where those sections, those twisted and folded sections come together and hold themselves in position with each other to form a specific three-dimensional shape. That's our tertiary structure, and then we also have quaternary structure, where two or more of those fully formed proteins will come together to form larger proteins. So that's the structure of proteins, and we talked about examples of proteins in the body, so from tendons and ligaments and all of our connective tissue, that's all proteins, but on a cellular level we have structural proteins. We also have proteins doing more sort of enzymatic kind of things, working as digestive enzymes or working as hormones and things like that. So what is your homework for this section? It is to build a protein. So what I'd like you to do is use something you have around the house. So it could be paper clips, it could be macaroni, it could be beads, it could be clay, it could be Play-Doh, it could be children, it could be, whatever you have. It could be blocks, and put them together. And what I want you to do is take a few pictures of it. So I want you to show me the primary structure, and then I want you to show me the secondary structure of it, and then show me the tertiary structure. So you'll need to build it until you can form all those layers of structure, and then even quaternary structure. And even if for quaternary structure you say, well, if I had 3 of these that you can see the tertiary structure in, and I put them together, that would be the quaternary structure. So if that doesn't work well for you to physically put together something like that and take pictures of it, you certainly could draw it by hand or draw it digitally and show me those layers of structure in a protein. And then you can take pictures of that and email it to me or text it to me, and that is your homework for this section. So next up we have the video on lipids, and then after that we have the video on ATP and DNA and RNA, and then that wraps up our organic chemistry section, and we'll have another test coming up very soon. Okay, so I will see you on the next video. Actually, I really will see you in class, and I hope this was enjoyable.